Hi everyone, in this video we are going to be talking about protein synthesis, and I want to start by thinking about a question that's really underlying this topic of protein synthesis, and that question is how you get from a gene to a trait. In the past couple weeks we've been talking about inheritance and thinking about how a person or an organism's genotype corresponds to their phenotype. And remember that a genotype is the combination of alleles that an organism has. So you can think about it as the specific versions of a gene that they might possess, and then how that dictates what their phenotype is going to be, what sort of traits they have. So when we're thinking about how you get from a gene to a trait, what we're really asking is how does DNA dictate what sorts of traits we have. Obviously, DNA doesn't dictate all the traits we have, but there are certain traits that are determined by our genes. So for those traits, how does that happen? And this process of protein synthesis, the topic for today's video lecture, is really understanding how proteins are made. And on one end, as sort of the starting point, we're going to have DNA. We're going to go through an intermediate molecule called RNA, and then finally we'll get to protein. So the whole topic of protein synthesis is what are all the steps that are involved in going from DNA to RNA to protein. And this video lecture will just be sort of a brief overview of sort of the big picture steps that are going to be involved with this. And then we'll have two more video lectures after this to follow up. One for going from DNA to RNA, and one for going from RNA to protein. So before we get started with this more big picture overview video, I just want to refresh our memory about a couple of things we already know that we've already covered so far this year, just so that we have the right things refreshed in our memory. So it's been a while since we talked about proteins, but hopefully you remember, or at least this video lecture makes you remember, that when we're thinking about a protein, remember our proteins are going to be these complex 3D shaped molecules that are going to be doing a bunch of different things in our body. And they're made up of polypeptide chains. And these polypeptide chains are in turn composed of amino acids. So we were saying that the amino acid is a monomer for the polymer, which is the protein. So amino acids are sort of the building blocks of the polypeptide chains, which then fold up into proteins. We also know that proteins are made by ribosomes. So ribosomes are sort of the protein-making factories of the cell. And then for the genetic side of things, we also know, and this is something we covered a little bit more recently, that genes are specific chunks or segments of DNA that are responsible for the phenotype or the traits of an organism. And when we're looking inside of a nucleus, we're going to see that there's DNA in the form of different chromosomes. So we know humans have 46 chromosomes. And if you sort of zoom in on the chromosome, you'll see that double helix shape of a DNA molecule. And so these genes contain the information that's needed to build a protein. So the information is here in the DNA, and we're going to use it to build these proteins. And all of this DNA, like I said, is in the nucleus of a cell. Okay, so here's a bird's eye view of what the process of protein synthesis is going to look like. And this is such a fundamental idea that it's sometimes referred to as the central dogma of molecular biology. So here's what the central dogma is. There's sort of three main steps. The first one is that a gene is going to be transcribed into RNA in the nucleus of the cell. Remember, the DNA lives in the nucleus, so this first step sort of has to happen where the DNA is, which is in the nucleus. And we'll go into more detail about what what RNA is. What does it mean to say it's transcribed? We'll go into more detail about each of these steps later, but this is just sort of a big picture. But basically, at this first step, we're making a copy of the DNA, but we're using a slightly different molecule called RNA instead. That's the first step. And then the second step is that the RNA is going to have to leave the nucleus and travel to the cytoplasm. The ribosomes don't exist inside of the nucleus, and the ribosomes are where proteins are built. So our sort of intermediate step is that we have to go from the nucleus, where the DNA is, to the cytoplasm where the ribosomes are. So the RNA is going to travel to the cytoplasm. And then the third step is that the RNA molecule is going to be translated into an amino acid sequence at a ribosome. So we're sort of converting this RNA molecule into those amino acid building blocks of a polypeptide. And then once this polypeptide is finished, it can be folded up into a protein. So those are the three main steps of protein synthesis, and they're referred to as the central dogma. And there are two processes that I've just described here. This first one, where we're going from a gene or a DNA into RNA, is called transcription. And then this last process, where we're building an amino acid chain out of that RNA, is called translation. So there's one more slide in this PowerPoint or in this video lecture, and then the following two video lectures, one will be dedicated to transcription and one will be dedicated to translation. So with this last slide, though, I just want to take a minute and sort of tell you a little bit more about this RNA molecule, because as you can see, it's sort of the thing that ties this whole process together. In the first step, we're going from DNA in the form of a gene, and we're making RNA. And then that RNA is being used to build amino acids. So we're going from DNA to amino acid, and the RNA is sort of the middle piece that's allowing that to happen. So what is RNA? RNA is a type of nucleic acid, and it's very, very similar to DNA, which we already know a lot about. The name RNA it's for ribonucleic acid, whereas we knew that DNA stands for deoxyribonucleic acid. And the reason for these differences in names has to do with the sugar that you can find along that sugar phosphate backbone. For DNA, the sugar that's in the sugar phosphate backbone is called a deoxyribose sugar, whereas in RNA, we have a ribose sugar. And really, literally the only difference is that it's still a carbon sugar that looks like this, and you can see for ribose, there's an OH down in this corner, whereas in deoxyribose, it's just an H. And so this deoxyribose it really means like without that oxygen, deoxy, whereas this one has an extra oxygen. So you don't need to know the details about the molecular structure of the sugar in the backbone, but just know that that's the reason that DNA and RNA have these different names. So the difference in sugar is one thing. Another main difference is that DNA, remember, has that double helix structure. It's got two strands, whereas RNA is usually found in a single-stranded form. So single-stranded RNA versus double-stranded DNA. And then the final difference is that the bases that we can find in DNA and RNA are slightly different. Three of the bases are going to be exactly the same. So we've got cytosine, we've got guanine, and we've got adenine, and those are the same for both DNA and RNA. But remember, we had T in DNA. It was A and T and C and G. So thymine is what we find in DNA, and RNA doesn't have thymine at all. Instead, it's got uracil. So whatever would be a thymine in DNA is going to be uracil instead in RNA. So the four bases for RNA are going to be cytosine, guanine, adenine, and uracil. The pairing is still going to be the same. So remember, cytosine and guanine are sort of paired up together. And then we had adenine and thymine that used to be paired up in DNA, or that are paired up in DNA. And adenine and thymine, which are paired up in DNA. A and T's go together, right? So in RNA, it's going to be the same. Cytosine and guanine will still be complementary to one another, and they'll be paired up. But instead of adenine having a pairing with thymine, it'll be paired with uracil instead. So adenine and uracil are the pair for RNA. Okay, so I'm going to stop this video here, and there will be another video following this one. The first one will be about transcription, and then the next one will be about translation. So I'll see you guys soon.